Hello, my inquisitive friend, and a very warm welcome to the Deep Filled channel. Tonight we will gently explore the most intimate corner of the cosmos you have ever known, that quiet yet vibrant space pulsing within your own skull, the human brain, this living architecture of tissue and electricity that never truly rests, even when you finally allow yourself to drift into stillness. And I promise to guide you through it all with such calm clarity that sleep may find you long before every question is answered. Before we wade into this captivating mystery, I would truly love to hear in the comments which corner of the earth you are listening from tonight. And if drowsiness is already tugging at you, simply leave a quiet hello. It might seem like a small gesture, but it signals to the platform that our space is a welcoming haven, helping us guide more weary minds toward relaxation alongside us. If you enjoy these scientific journeys, please leave a like and subscribe. And to those already part of our community, my deepest gratitude. If you wish to draw even closer, consider joining our channel membership. Members enjoy early access to every video before anyone else. You will find the link waiting in the description below. Now, wherever you happen to be, settle into a truly comfortable position. If sleep begins to call, simply accept its invitation. Let us begin. Something is unfolding inside you right this very moment that you never consciously chose. Your eyes are absorbing these words, or your ears are translating these sounds, and meanwhile, another part of you is quietly regulating your body temperature, monitoring the pressure within your blood vessels, adjusting muscle tension to keep you exactly where you are, and simultaneously crafting the mental state we call relaxation or focus or drowsiness. All of this unfolds at once, without you needing to direct any of it. You never had to learn how to make your heart beat. You never studied how to breathe while asleep. You never memorized how to widen your pupils when the light fades. Something within you simply knows. And that quiet intelligence is what we will explore with gentle patience tonight. Recall the most recent dream you experienced. It might have been bizarre, fragmented, filled with unfamiliar faces that somehow felt known. Perhaps it was a dream of falling or flying or wandering an endless hallway. You never planned that dream. No conscious part of you wrote its script, yet those images arrived with a vividness that sometimes rivals waking life. Who crafted them? Now bring to mind a childhood memory, the aroma of a meal your grandmother prepared, the feel of a fabric you have not touched in decades. Where did it surface from? How can something that occurred so long ago still reside within you with enough clarity to stir a genuine emotion? Your brain preserved it, not as a file in a folder, not as a recording on a tape, but in a far stranger, far more fluid, far more remarkable way than any technological analogy could ever capture. Most people move through their entire lives without ever truly examining the organ that makes every single moment of that life possible. We use the brain to contemplate everything except itself. It is like using your eyes to observe the entire world while never pausing to consider the very mechanism of sight. And what is most astonishing is that everything you are feeling right now, the gentle weight lifting from your shoulders, the way your breathing gradually deepens, the sound of my voice softening into something more like a texture than mere words, all of this is your brain interpreting the world and constructing, in real time, the experience you call reality. There is a foundational truth about the brain that overturns nearly every popular assumption we hold about it, and this is where we must begin, because this realization shifts everything that follows. The truth is this: the brain does not record the world exactly as it exists. It builds the world as it might be, using fragmented pieces of sensory data, and then presents that construction to you as though it were direct reality. You are not seeing what is literally in front of you right now. You are witnessing your brain's interpretation of what lies before you, an interpretation woven from past memories, expectations, emotional states, and a vast number of unconscious assumptions. The world you experience is, in a very real sense, a remarkably well-tuned hallucination. This might feel unsettling at first glance, but as you settle into the idea, it becomes far more fascinating than frightening. It reveals that the brain is not a passive mirror reflecting reality. It is an active architect of experience. Every time you look at something, the brain does not merely capture an image like a camera. It takes the electrical signals arriving from your eyes, cross-references them with everything it has seen before, fills in the missing pieces, makes educated guesses about what is likely there, and generates something that feels like direct perception. There is a blind spot in each of your eyes, a location where the optic nerve meets the retina and no light-sensitive cells exist. You have never noticed this gap because your brain seamlessly paints over it with what it expects to be there, without warning you, without asking permission, without leaving any visible trace of its quiet intervention. This process occurs with everything, with sounds, with flavors, with the very sensation of your body occupying space. The brain is constantly editing, completing, and interpreting, and it accomplishes this with a speed and precision that no machine humanity has ever built has even come close to matching. Let us now turn to the physical stage where all of this unfolds, because before we can grasp what the brain does, we need some sense of what it actually is. The average adult human brain weighs roughly 1.5 kilograms. To put that in perspective, it is about the weight of a large avocado or a compact laptop. Yet no avocado and no laptop has ever composed a symphony, or felt longing, or feared mortality, or laughed at a joke. Weight tells us very little about capability. What truly matters is not mass but organization. And by any reasonable measure, the human brain is the most intricately organized system we have ever encountered in the observable universe. Its texture is often compared to firm tofu, or soft gelatin, or a dense paste. It lacks the rigid solidity we might imagine when thinking of something as seemingly permanent as our own mind. If you were to lift it from the skull, it would yield gently to the touch. It must float in a protective fluid, the cerebrospinal fluid, to avoid being crushed by its own weight against the inner walls of the cranium. This fluid acts as a cushion, a transparent shock absorber that softens the impact of every step you take, every time you lie down, every subtle tilt of your head throughout the day. From the outside, what comes to mind when you picture a brain are those distinctive folds and grooves, the wrinkled ridges that resemble a giant walnut or a landscape shaped by millennia of gentle erosion. These folds have a name, gyri, and they exist for a beautifully practical reason: to maximize surface area within a confined space. If you were to carefully unfold the cerebral cortex, the brain's outermost and most complex layer, and lay it completely flat, it would cover roughly 2,500 square centimeters, about the size of a large sheet of wrapping paper. Yet that very same material has been intricately folded and compressed to fit inside a skull that rests comfortably in your hands. Nature is an extraordinary engineer of compact design. Beneath and woven through the cortex lies a vast expanse of pale tissue known as white matter, which stands in contrast to the gray matter of the cortical surface. This white matter consists of trillions of connecting fibers, long cables that link different brain regions to one another, much like the wiring inside a building connecting room to room, floor to floor. The pale color comes from a fatty substance called myelin, a protective sheath that wraps around these cables like the insulation around an electrical wire, dramatically increasing the speed at which electrical signals travel from one destination to another. Yet the true protagonist of this story, The essential element without which nothing we have described could exist is the neuron. The human brain contains approximately 86 billion neurons. 86 billion. That number is so vast it requires a concrete image to feel real. If you counted one neuron per second without pausing, without sleeping, without eating, it would take you nearly 2,700 years to reach the end. You would have begun counting before the birth of Christ and would finish somewhere far into our future. And neurons are only one piece of the landscape. Alongside the neurons reside glial cells, which for decades were dismissed merely as the brain's glue, as if they were nothing more than structural filler with little real purpose. Modern science has thoroughly dismantled that myth. Glial cells are active, essential participants in brain function, involved in shaping connections between neurons, clearing metabolic waste, regulating electrical communication, and possibly even influencing thought and memory in ways we are only beginning to grasp. There may be as many glial cells as neurons, or perhaps even more, and to overlook them would be like describing a forest by mentioning only the trees while ignoring the soil, the fungi, the insects, and the entire invisible network that sustains life there. Each neuron is, in itself, a structure of astonishing elegance. Picture a cell with a central body, the soma, from which branch-like extensions spread outward. These branches are called dendrites, and their role is to receive signals from neighboring neurons. Extending from the cell body is also a single elongated projection, the axon, which acts as a transmission cable carrying electrical impulses away, sometimes to nearby cells, sometimes to cells on the opposite side of the brain, or even down into the spinal cord. The tip of the axon splits into terminals that approach, but never quite touch, the dendrites of other neurons. That infinitesimal gap between one neuron and the next is called a synapse, and it is there in that tiny void that the true magic of cerebral communication unfolds. When a neuron fires, it sends an electrical impulse racing down the axon to the synaptic terminals. Upon arrival, that impulse triggers the release of chemical messengers, known as neurotransmitters, which drift across the synaptic gap and lock into specific receptors on the receiving neuron. This binding works like a key sliding into a lock. When it occurs, it can either excite the next neuron, prompting it to fire as well, or inhibit it, making it less likely to activate. The outcome of every thought, every emotion, every memory, every movement is the collective pattern of thousands or millions of these chemical conversations unfolding in fractions of a second. Dozens of different neurotransmitters exist, each carrying distinct and often overlapping roles. Dopamine participates in the reward system, in motivation, in the anticipation of pleasure. Serotonin shapes mood, appetite, and sleep cycles. Glutamate serves as the nervous system's primary accelerator, the main excitatory signal. Gamma-aminobutyric acid, known as GABA, acts as the primary inhibitor, the brake that keeps the brain's electrical activity in careful balance. Norepinephrine prepares the body for action. Acetylcholine supports attention, learning, and muscle activation. This chemical orchestra never falls silent. Right now, as my voice grows softer and your body settles more deeply into wherever you are resting, these molecular messengers are dancing within your skull, precisely calibrating how much drowsiness your brain will allow to wash over you. There is a persistent myth that deserves to be gently set aside precisely because it has survived for so long despite being entirely inaccurate. It is the notion that human beings only utilize 10% of their brains. This claim has circulated for generations, appearing in films and advertisements, and it feels intuitively appealing because it hints at a vast hidden potential waiting to be unlocked. Yet it holds no grounding in actual neuroscience. Brain imaging techniques such as functional magnetic resonance imaging, consistently demonstrate that virtually every region of the brain becomes active at some point, and that even during simple tasks, enormous networks of neurons light up simultaneously. During deep sleep, when one might reasonably expect the brain to power down, it remains highly active, consolidating memories, clearing metabolic byproducts, and reorganizing connections. There is no silent reserve waiting to be discovered. Another misconception that deserves to be quietly released is the belief that the brain is permanently split between a rational side and a creative side, with the left hemisphere housing logic and language, and the right hemisphere serving as the home of intuition and artistry. This division was extrapolated from genuine observations, but stretched far beyond what the data actually supports. It is true that certain functions tend to be more dominant in one hemisphere than the other. Language production, for instance, is more commonly localized in the left hemisphere for most individuals, but the idea that two separate personalities reside inside your skull, one analytical and one artistic, operating somewhat independently, does not align with contemporary neuroscience. The two hemispheres are in constant dialogue through a thick bridge of nerve fibers called the corpus callosum, and most complex cognitive activities involve distributed networks that integrate both sides seamlessly. The brain is also not a uniform structure. It is organized into distinct regions with different specializations, yet they always operate in concert. The cerebral cortex, that wrinkled outer layer we mentioned, is divided into lobes, each with its own functional tendencies. The frontal lobe, situated at the front, handles planning, decision-making, impulse control, personality, and the ability to imagine the future. It is the region that most distinguishes us from other animals, and also the last to fully mature, a process that extends well into the early 20s. The parietal lobe, resting toward the top and back, integrates sensory information, processes spatial awareness, and contributes to attention and the construction of the boundary where your body ends and the world begins. The temporal lobe, located on the sides, is deeply involved in hearing, language, facial recognition, and, crucially, memory, particularly through an inner structure called the hippocampus, shaped like a seahorse, which is essential for forming new memories. The occipital lobe at the rear is primarily dedicated to visual processing, and it is there that signals arriving from your eyes are transformed into images that feel as though they exist out in the world, but as we now know, actually reside within. Deeper within the brain, beneath the cortex, lie evolutionarily older structures that humans share with other mammals and even reptiles. The limbic system, a collection of structures including the amygdala and hippocampus, governs emotions, emotional memory, fear, and pleasure. The amygdala, roughly the size of an almond, is renowned for its role in fear responses, acting as the structure that rapidly evaluates whether something in the environment poses a threat before your conscious mind has even finished processing the situation. The hypothalamus, even smaller, regulates fundamental functions like hunger, thirst, body temperature, sleep-wake cycles, and serves as the bridge between the nervous system and the endocrine system, the network of glands that produces hormones. Further back and downward sits the cerebellum, which many overlook when thinking about the brain, yet it contains roughly half of all the neurons in the central nervous system, packed into a relatively compact structure. The cerebellum is vital for motor coordination, balance, and movement precision. When you lift a cup without spilling, when you walk without consciously planning each step, when you play an instrument or dance, the cerebellum is working quietly in the background, a discrete conductor ensuring the orchestra of movement stays perfectly in tune. And finally, there is the brainstem, the oldest and most fundamental region, which connects the brain to the spinal cord and controls the most essential survival functions: breathing, heart rate, blood pressure, sleep, and wakefulness. It is the part of the brain that never sleeps, never takes a break, and works from your first breath to your last without asking for recognition or gratitude. All of this entire breathtaking architecture of lobes and structures and pathways and synapses fits inside a skull. It fits inside you. And right now, as you allow these words to dissolve into something closer to a sensation than to information, this entire system is sustaining your presence in the world, regulating every process, maintaining the delicate balance between being awake and being asleep, between listening and dreaming. Which brings us to a question that will linger in the air as you grow slower, quieter, heavier. This entire system, 86 billion neurons, trillions of synapses, the infinite orchestra of electrical signals and chemical messengers, consumes an amount of energy that would be staggering for such a small organ. But where does that energy come from? How can an organ weighing less than 2 kilograms sustain this continuous activity day and night without pause? The answer to that question is not simple, and it touches something deeper than just the biochemistry of cerebral metabolism. It leads us to understand why the brain is so demanding, so carefully shielded, so reliant on a supply that can never be interrupted, and what occurs when that supply begins to fluctuate, as it is doing now, gently, as sleep draws nearer. Imagine a candle burning steadily on a table. It consumes wax, transforms matter into light and warmth, and as long as there is enough wax, it burns quietly and consistently. Now imagine that this candle can never be extinguished, that it must keep burning while you sleep, while you dream, while your heart beats slowly in the quiet hours of the night. It must burn even when everything around you is in deep rest. That candle exists inside your head, and it is fueled by a substance so simple that you likely encountered it today at breakfast, in bread, in fruit, in rice. That substance is glucose, and the brain consumes it with a dedication unmatched by any other organ in the human body. Glucose is a simple sugar, a small and elegant molecule that the digestive system extracts from food and releases into the bloodstream. From the blood, it travels throughout the body, distributed as a resource that every cell can use to generate energy. The liver stores it in temporary reserves, muscles utilize it during exertion, organs consume it in proportion to their size and activity. But the brain has a different relationship with glucose, a relationship that borders on absolute. While muscles can, in times of need, switch to fat as an alternative fuel, and while the heart can function reasonably well with various energy substrates, the adult human brain relies on glucose almost exclusively. It represents only about 2% of total body weight, yet it consumes roughly 20% of all the glucose the organism processes. It is as if a tiny component of a massive engine demanded 1/5 of all available fuel simply to keep existing. This consumption is not accidental. It reflects an architecture built over millions of years of evolution that bet on a high metabolism organ as the solution to behavioral complexity. For 86 billion neurons to maintain their electrical potentials, for trillions of synapses to release and reabsorb chemical messengers, for cell membranes to continuously pump ions in and out, creating that permanent electrical whisper described earlier, all of this requires energy. And that energy comes from a process occurring inside every neuron, within microscopic structures called mitochondria, which exist in surprisingly high numbers in nerve cells. Mitochondria take glucose, or rather a derivative of it called pyruvate, and through a chain of chemical reactions produce a molecule called adenosine triphosphate, which functions as biology's most fundamental energy currency. Each neuron uses this adenosine triphosphate to perform the work of being a neuron. What that work actually means is fascinating to contemplate. When a neuron fires an electrical signal, it allows sodium ions to rush quickly through its membrane, depolarizing it, creating that wave of activity that travels down the axon like a spark. Immediately afterward, it must pump those ions back out, restoring its resting state so it can fire again. This pumping is not passive, it demands energy. Specific proteins in the neuron's membrane, called sodium-potassium pumps, function like tiny molecular machines, and each cycle of these machines consumes adenosine triphosphate. It is estimated that maintaining the ionic balance of a single resting neuron already consumes a significant portion of its total energy production, and when the neuron is active, that consumption rises even further. Multiplied by 86 billion cells, this number becomes something the human brain experiences as a demand that never ceases. This is why the blood supply to the brain is treated with a priority no other organ receives. The heart pumps blood throughout the body, but there is an automatic regulation system in cerebral vessels called cerebrovascular autoregulation that ensures blood flow to the brain remains steady even when blood pressure fluctuates. If you stand up quickly and your pressure drops momentarily, cerebral vessels dilate to compensate. If your pressure rises, they constrict to prevent excess flow from causing damage. The brain protects its glucose supply with a regulatory sophistication that took physiologists decades to fully understand. The loss of consciousness that occurs when this system briefly fails, that moment of dizziness or vision darkening some people experience when standing suddenly, is a direct demonstration of how much conscious functioning depends on an uninterrupted flow. And here we arrive at a turning point in the history of brain science, a moment led by a man named Seymour Cady, an American neuroscientist who worked in the latter half of the 20th century and developed the first methods to accurately measure how much glucose the human brain consumes and exactly where that consumption occurs. Before Cady, the question of cerebral metabolism was largely speculative. It was known that the brain used energy, that it required blood and oxygen, that sudden deprivation caused loss of consciousness within seconds. But there was no way to measure this precisely in a living human. Cady developed a technique using traceable substances in the blood entering and leaving the brain, allowing researchers to calculate the difference between what arrived and what departed, and thus determine the net consumption of oxygen and glucose by brain tissue. It was a technical breakthrough that opened a new window into the body's most inaccessible organ. What Cady discovered both confirmed and deepened the mystery. The resting brain, the brain of a relaxed adult closing their eyes and simply existing, already consumed an enormous amount of energy. But what happened when that adult began thinking intensely, solving a complex mathematical problem, or recalling a powerful emotional memory? Total consumption increased very little. This counterintuitive result led to an important refinement in understanding. The brain does not function like a muscle that uses significantly more energy when working harder globally. It redistributes its energy. When a specific area becomes more active, local blood flow to that region increases, but other areas may slightly reduce their activity. Total consumption remains surprisingly stable, but the internal pattern is constantly shifting, a living map of higher and lower activity hotspots that never repeats in exactly the same way. This discovery planted the seed for what would later become functional magnetic resonance imaging, the technique that now allows neuroscientists to observe the living human brain while it thinks, feels, decides, and remembers. Functional MRI does not measure electrical activity directly. It measures changes in local blood flow based on the ratio between oxygenated and deoxygenated hemoglobin, a signal called BOLD, which is essentially a metabolic footprint of neuronal activity. Every time you see colorful brain images in scientific papers, with areas glowing in red and yellow indicating activity, what you are actually seeing is essentially a map of local energy consumption, a direct descendant of Kitty's pioneering investigations into cerebral metabolism. But there is an even more fundamental law governing all of this, a law that goes beyond biochemistry and touches the most basic physics of any system that processes information. It is the second law of thermodynamics, and it states in its simplest essence that any process transforming energy from one form to another produces heat as a byproduct, and that this heat represents a disorder that cannot be completely reversed. Every time a sodium-potassium pump pushes ions against their concentration gradient, every time a synaptic vesicle releases its neurotransmitters into the gap between two neurons, every time a mitochondrion converts glucose into adenosine triphosphate, a portion of the available energy is converted into heat that dissipates into the environment. The human brain, therefore, generates a considerable amount of heat. Part of the warmth you feel inside your head, especially after a period of intense concentration, comes directly from neuronal metabolism. This thermodynamic law carries an implication that extends far beyond heat. It implies that information processing has an irreducible physical cost. There is no free computation. There is no thought without an energy consequence. Every memory formed, every decision made, every emotion felt leaves a trace in the universe's energy balance in the form of increased entropy, of disorder that cannot be undone. The physicist Rolf Landauer formulated this mathematically in the mid-20th century by showing that erasing a single bit of information necessarily releases a minimum amount of heat. The brain does not erase bits the way a computer does, but the principle applies in a broader sense. The work of creating, maintaining, and transforming neural representations carries a thermodynamic cost. Thought literally warms the world on a minuscule but real scale. There is another physical law that governs brain function in an even more direct way, and it comes from electrodynamics. It is the law that describes how electrical charges move, how electric fields propagate, and how currents are generated and dissipated. Neurons are essentially conductors immersed in a conductive medium, the extracellular fluid, and when they fire, they generate electric fields that spread through brain tissue, following the equations of electromagnetism. With the same precision, those equations describe electricity in a copper wire. This is why an electroencephalogram works. Electrodes placed on the scalp detect the electric fields produced by the synchronized activity of millions of neurons in the superficial layers of the cortex. Synchronization is crucial here, because a single neuron produces an electric field so tiny it could never be detected from outside the skull. But when many neurons fire together in coordinated patterns, their fields sum together and create waves that can be measured, recorded, and analyzed. Hans Berger was the man who first recorded these waves in human beings. He was a German psychiatrist working in the early 20th century, and his story carries something both quixotic and deeply moving. Berger was convinced that a connection existed between the brain's electrical activity and mental phenomena, that somehow cerebral electricity was the physical signature of thought. For years he worked in relative isolation, building his own equipment, refining his techniques, enduring the skepticism of colleagues who doubted it was possible to measure something so subtle through the skull and scalp. In 1929, he published his first recordings of the human electroencephalogram, showing rhythmic oscillations he called alpha waves. Waves of about 10 cycles per second that appear when a person is relaxed with closed eyes and disappear when they open them or begin thinking actively. Berger interpreted alpha waves as an organized resting state of the brain, a kind of coordinated idle rhythm the organ adopts when it is not intensely processing the external environment. This interpretation was fundamentally correct, and alpha waves remain one of the most widely used markers in sleep and meditation research and clinical practice to this day. When you close your eyes and allow thoughts to dissolve, when the objects around you stop demanding your attention and your mind begins to drift. Your electroencephalogram would likely show exactly what Berger described nearly a century ago, that gentle rhythmic oscillation, that electrical signature of the brain in attentive rest. What Berger could not have imagined was that other rhythms existed, slower and deeper, that only appear during sleep. Delta waves, oscillations of fewer than 4 cycles per second, emerge during the deepest phases of slow-wave sleep and represent a large-scale synchronization of the cortex that has no equivalent in the waking state. During these waves, vast populations of cortical neurons alternate between periods of near-complete silence and bursts of activity in a rhythm that contemporary neuroscientists understand as fundamental for memory consolidation and the restoration of cerebral metabolism. Deep sleep, in other words, is partly an opportunity for the brain to balance its energy accounts so that cleaning and restoration processes can operate without the constant demand of processing information from the outside world. This brings us to a fundamental imbalance that makes the brain function, the imbalance that lies at the root of all its activity, of all its operational beauty. Neurons exist in a permanent state of tension between two poles. There is a constant tendency for the ionic gradients that sustain the membrane's electrical potential to dissipate, for ions to move according to their concentration gradients and eliminate the charge difference that is the prerequisite for every nerve signal. This tendency is physical, inevitable, dictated by the laws of diffusion and electrochemistry. And there is a constant effort to counteract this tendency, carried out by ionic pumps consuming adenosine triphosphate, restoring the gradients, maintaining the possibility of the next firing. The neuron exists between firing and resting, between order and dissipation, sustained by the relentless work of metabolism. This imbalance is not a design flaw, it is the design. A neuron in perfect equilibrium with its environment would be a dead neuron, a neuron that can no longer fire because it has lost the necessary potential difference. The life of a neuron is the active maintenance of imbalance, the continuous effort against the thermodynamic tendency of everything to equalize. And this struggle happens in each of the 86 billion cells in your brain, right now as you rest. They are all there, maintaining their tensions, consuming their energy molecules, waiting for the next signal, or sending the next signal or receiving the next signal from their neighbors. There is something deeply analogous to this across many scales of nature. A river is the result of an imbalance between the altitude of a mountain and sea level, and it exists as long as that imbalance exists, flowing, shaping its bed, carrying sediment, feeding ecosystems. When the imbalance resolves, when all the water finds its level, the river stops. The neuron is a molecular river, sustained by the ionic concentration difference between inside and outside, flowing from potential to firing and back to potential, as long as metabolism ensures the gradient is restored after each descent. But there is an additional layer to this story that makes neuronal imbalance especially fascinating. Neurons do not work alone. They are connected in networks where the imbalance of one can trigger the imbalance of another, where the firing of one neuron can excite or inhibit its neighbors, where activity patterns propagate and transform into processing cascades that emerge from local interactions but produce global phenomena. Consciousness, thought, memory, perception, all these phenomena you associate with what it means to be you emerge from interactions between neurons that individually know nothing about the world. This is where the story of energy meets the story of information and where cerebral metabolism reveals its true meaning. Glucose is not merely keeping neurons alive, it is making possible the creation of patterns, the difference between signal and noise, the distinction between a neuron that fired and one that did not, which is the most fundamental unit of information in the nervous system. Every molecule of adenosine triphosphate spent on ionic pumping is a molecule spent creating the conditions for information to exist. Cerebral metabolism is, at its deepest essence, the physical cost of the possibility of thought. And when that cost begins to be managed differently, when the body starts preparing the ground for sleep, when core temperature begins to drop slightly, when certain vigilance systems reduce their activity, when those slow oscillations begin to appear in electroencephalographic recordings, something shifts in how neurons manage their fundamental imbalance. Some networks remain active, others begin to synchronize into slower rhythms. The pattern of glucose consumption redistributes. The candle does not go out, but its flame changes shape, color, intensity as if it were burning with a different intention. Now picture a single neuron in some deep region of your cortex. It receives signals from perhaps thousands of other neurons. Each signal arrives at the synapse, releases its neurotransmitters, opens or closes ionic channels, tilts the membrane's balance slightly toward firing or slightly toward inhibition. The neuron integrates all these influences across its membrane and in a fraction of a second decides if that word can be used for such a physical and automatic process, whether the sum of influences has crossed the threshold that triggers an action potential. If it has, an electrical wave races down its axon, reaches its synaptic terminals, releases neurotransmitters to the next neurons in the network, and the cycle begins again elsewhere. If it has not, the neuron remains silent for that moment, and the influence that arrived dissipates without consequence. This mechanism, so elegant and so physical, happens not in one neuron but in 86 billion of them, in parallel, continuously, each receiving and sending and integrating and firing or not firing in a tissue where connectivity between cells creates combinatorial possibilities that surpass any number the human mind can intuitively grasp. And a question begins to float as gentle as the very thought it describes. What happens when this simple mechanism of receiving, integrating, and firing repeats not once, not 1,000 times, but trillions of times per second, in networks that have learned over years to organize their activity patterns in specific ways to preserve some configurations and discard others, to build upon the physical substrate of ionic imbalance and glucose metabolism, something that goes far beyond electrical signals in lipid membranes. When trillions of local events occur simultaneously in the same tissue, something begins to emerge that was not contained in any of the individual events. It is not a sum, it is not an average. It is a new quality, an organization that arises between the parts and not within any of them, as if the very space between neurons were capable of storing an intention that no isolated neuron could ever carry. This emergence is the principle that governs everything the brain does at a large scale, and understanding it requires the mind to step back slightly from the image of individual cells firing and begin to see nervous tissue as a field, as an ocean of possibilities that organizes itself into currents, into eddies, into patterns that have their own internal coherence. The first level of this emergent organization begins in something neuroscientists call a cortical column. Imagine a microscopic cylinder of tissue perpendicular to the cortical surface, containing between 100 and a few hundred neurons stacked in distinct layers. This cylinder is the basic functional unit of the cerebral cortex, the smallest structure that can already be said to process information in a relatively autonomous way. Neurons within a column are connected to each other by far denser connections than the connections each maintains with neurons outside the column. This internal density creates a kind of preference, a tendency for the electrical activity of one neuron to propagate first to its closest neighbors before reaching more distant structures. Neuroscientist Vernon Mountcastle was the first to describe this arrangement clearly in the late 1950s by observing that neurons arranged along the same vertical axis all responded to the same type of stimulus, while neurons arranged horizontally, at the same depth but in different lateral positions, could respond to completely different stimuli. This discovery was a moment of quiet and profound insight. The cortex was not a homogeneous field, it was a mosaic of modules, each module with its own specialization, and the vertical geometry of the neuronal stack was what created that specialization. But columns do not exist in isolation. They cluster into regions, and regions organize according to a geographical principle anatomists call a topographic map. In the cortical region that processes vision, neurons are arranged such that neighbors in the eye's visual field correspond to neighbors in the brain tissue. It is not a perfect correspondence, not a photograph, but it is a continuity. Adjacent points in the visual world are represented in adjacent points in the cortex. The same occurs in the somatosensory cortex, which processes touch. Each region of the body is mapped onto a specific strip of cortex, such that the thumb and index finger are represented in adjacent cortical regions, even though the entire hand occupies an area disproportionate to its physical size, because the density of sensory receptors in the skin of the fingers is much higher than the density of receptors in, say, the back or the thigh. This distorted map, where lips and hands and tongue occupy vast cortical territories, while the trunk and legs are compressed into narrow strips, has a name given with a certain poetic irony by neurosurgeon Wilder Penfield: the cortical homunculus, a little imaginary man whose body reflects not real anatomical proportions, but the functional importance of each part. Penfield arrived at this map not by accident but by clinical necessity. During surgeries to treat severe epilepsy, it was necessary to identify which cortical regions were responsible for which functions before removing diseased tissue, to avoid causing permanent damage. With the patient awake under local anesthesia, Penfield electrically stimulated different points of the cortex and asked what the person felt: tingling in the thumb, a sensation of movement in the shoulder, a smell that recalled childhood. The figure that emerged from these conversations inside operating rooms over decades of work was that of the brain as an atlas of itself, each region not only processing information but being, in a certain way, the territory it represents. Seeing with the occipital cortex is, from this perspective, something like being the visual world. Feeling touch in the somatosensory cortex is being the surface of the body. The separation that common sense imagines between the organ that perceives and the world that is perceived begins to dissolve in this description, and in its place remains something more subtle and more unsettling: the idea that the brain does not receive the world, it reconstructs it region by region, map by map, in living tissue. These maps are not static. One of the most surprising discoveries of recent decades is that the topographic organization of the cortex is plastic, that it changes in response to experience, that the amount of cortical territory dedicated to a function can increase or decrease depending on how much that function is exercised. Musicians who have played string instruments for many years show an expansion of the cortical region dedicated to the fingers of the fingering hand. People who lose a limb and go years without receiving sensory signals from a certain body region eventually see that strip of the cortical homunculus colonized by neighboring maps, like a garden advancing into an abandoned plot. The phenomenon has a name: activity-dependent synaptic plasticity, but that technical expression does not capture the beauty of the process. What it describes is that the brain is a map that continuously redraws itself, that the geometry of its organization is the condensed history of everything the body has done and felt and practiced, that every centimeter of cortex is, in a certain way, an autobiography inscribed in tissue, Above the level of regional maps, the brain organizes itself into long-distance networks, groups of regions separated by centimeters or tens of centimeters of tissue establish high-speed connections through bundles of myelinated axons, the great white highways that run beneath the gray cortex and collectively form what anatomists call white matter. The whitish color of these bundles comes from myelin, the lipid sheath that wraps long axons and allows electrical signals to travel at speeds that can reach 120 meters per second, a speed that transforms what could be a debilitating delay into nearly instantaneous communication between distant regions. Without myelin, the electrical signal would lose strength along the axon, like a voice dissipating in open air. With it, the signal jumps from node to node along the axon, regenerating itself, maintaining its strength, arriving at its destination with the same intensity with which it departed. The distribution of these white matter bundles follows patterns that neuroanatomists spent centuries trying to chart. Some of these pathways are ancient in the sense that they appear in virtually all examined vertebrates and serve functions that existed long before the expanded human cortex. The corticospinal tract, for example, is a bundle that directly connects the motor cortex to the spinal cord, allowing voluntary and precise muscle control, and is one of the anatomical markers of sophisticated intentional movement capacity. The corpus callosum is an even more imposing bundle, a structure that connects the two cerebral hemispheres along almost the entire length of the cortex, allowing information processed in the left hemisphere to reach the right hemisphere in milliseconds and vice versa. Its surgical section, performed in extreme cases of epilepsy that did not respond to other treatments, produced some of the most revealing experiments in the history of neuroscience, when split-brain patients demonstrated behaviors suggesting two partially independent processing centers coexisting in a single skull, each with access to different fragments of experience, each capable of producing responses and even intentions the other was unaware of. The geometry of the two hemispheres and their imperfect mirror relationship is itself one of the most fascinating patterns of large-scale brain organization. The two sides are not identical. The left hemisphere is, in most people, dominant for language. The regions that produce and comprehend speech are concentrated in it asymmetrically, an organization that begins to outline itself during fetal development and is influenced by genetic and epigenetic factors scientists are still mapping. The right hemisphere is more specialized in processing global contours, spatial configurations, musical melodies heard as wholes rather than sequences of isolated notes. This division is neither absolute nor impermeable, and the corpus callosum ensures constant integration between the two sides, but the functional asymmetry of the hemispheres is real enough that damage to equivalent regions on both sides produces different deficits, that a lesion in Broca's area in the left hemisphere can completely suppress speech production, while an equivalent lesion in the right hemisphere may leave language intact but impair the ability to understand jokes or metaphors, to perceive emotional tone in a voice, to read between the lines. Beyond the hemispheres and their asymmetries, the brain organizes itself vertically into structures that reflect layers of evolutionary time. The distinction between cortex, limbic system, and brainstem is not merely anatomical, it is an archaeology, a stratigraphic record of the history of life with a nervous system. The brainstem, the innermost and oldest structure, controls functions that cannot wait, that cannot be delegated to slow and deliberative processing: heart rate, breathing, blood pressure, basic reflexes for orienting to sound and movement. Damaging the brainstem threatens life immediately, because it sustains the processes that keep the body alive regardless of what the cortex decides to do. Above the brainstem, the limbic system forms a ring of interconnected structures that mediate emotions, memories, and the impulses that direct behavior toward rewards and away from threats. The hippocampus, perhaps the most studied structure of the limbic system, is involved in forming new episodic memories, in recording events situated in time and space. The amygdala, an almond-shaped structure situated deep within each temporal lobe, evaluates the emotional significance of stimuli with a speed that precedes consciousness, such that the heart accelerates and muscles tense before the verbal mind has even formulated the perception that something is threatening. The hypothalamus, small but extraordinarily powerful, integrates hormonal and neural signals to regulate appetite, thirst, body temperature, sleep-wake cycles, reproductive behaviors, and an entire constellation of functions that keep the organism in balance with environmental demands. These different levels of the cerebral hierarchy communicate with each other through multiple pathways, and the nature of these communications is bidirectional in ways that continually surprise researchers. The prefrontal cortex, the most anterior region of the frontal lobe and one of the last to mature during human development, projects inhibitory and modulatory connections to the amygdala, allowing what is popularly called emotional regulation, the ability to recognize a threat without being completely overwhelmed by it, to feel fear without acting impulsively out of fear. But the amygdala also projects back to the prefrontal cortex, and its influence can, under conditions of intense stress or immediate danger, suppress the deliberative processing of the cortex, interrupting paused reasoning in favor of rapid and automatic responses. This gentle war between ancient structures and newer structures, between systems that prioritize speed and systems that prioritize accuracy, between impulse and reflection, is one of the most pervasive organizing principles of human behavior, and its expression in different situations determines much of what each person does, thinks, and feels throughout a lifetime. Above this level of individual structure analysis, neuroscientists of the latest generation have developed tools to observe the entire brain in function and identified something even more comprehensive: networks of regions that activate and deactivate together consistently over time, even in the absence of any specific task. The most famous of these networks is called the default mode network, and its existence was ignored for a long time simply because neuroimaging experiments focused on regions that activate during tasks and discarded as background noise the regions that become more active when the person is not doing anything in particular. When researchers finally turned their attention to this seemingly inert background, they discovered it was actually a highly organized and metabolically costly network of regions distributed across the medial prefrontal cortex, the posterior cingulate cortex, the parietal lobes, and the hippocampi, regions that worked in concert during daydreaming, during mental projection into the future, during imagining others' perspectives, during reflection on oneself. The discovery of the default mode network was disconcerting because it suggested that the resting state of the human brain is not passivity but a different kind of work, a form of processing turned inward, toward integrating past experiences with future projections, toward building and maintaining a narrative of identity. The brain does not wait for stimuli to arrive to begin working. It works continuously, and external stimuli are, from a certain perspective, interruptions of an internal processing that never stops. This idea inverted a fundamental intuition that had dominated neuroscience for decades, that the brain is a reactive system that waits for inputs to produce outputs, and replaced it with a much more dynamic view, that of a system that continuously generates hypotheses and predictions and uses sensory inputs less to describe the world than to correct the predictions it was already making. Other large-scale networks have been identified with similar methodologies. The salience network, involving the insular cortex and anterior cingulate, monitors the internal and external environment for changes that deserve attention, functioning as a sensitive relevance detector that directs processing resources where they are most needed. The frontoparietal network, also called the executive control network, is recruited for tasks requiring sustained attention, planning, and cognitive flexibility. The ventral visual network processes object identity, what things are, while the dorsal visual network processes location and orientation for action, where things are and how to interact with them. These networks do not operate in isolation. They interact, compete for resources, mutually suppress each other in some contexts, and coordinate in others, in a dance of activity patterns whose choreography is being slowly deciphered by researchers who have dedicated their careers to looking at functional MRI images and trying to extract from them the logic of the mind. What emerges from this multi-scale view, from the ion crossing the membrane to the network spanning both hemispheres and all lobes, is the image of a system that organizes itself according to principles of efficiency and specialization, while maintaining global connectivity that allows integration. Network theorists call this property a small world network, an arrangement where any node can be reached from any other node through a surprisingly small number of connections, even though most connections are local. The human brain appears to exhibit exactly this property, highly clustered locally, with dense modules of connections at a regional scale, but with enough long-distance connections that information can propagate rapidly throughout the entire system when necessary. This small world architecture appears to be the solution evolution found to the problem of processing information efficiently in an organ whose growth is limited by skull size and whose energy consumption is already disproportionately high compared to any other body tissue. Because the brain is extraordinarily expensive in metabolic terms, representing less than 2% of an adult's total body mass, it consumes around 20% of all the energy the organism extracts from food, a proportion that has no equivalent in any other organ. This energy demand imposes real constraints on the evolution of brain organization. Every new connection, every additional neuron, every extra layer of processing has a cost that must be justified by the functional benefit it brings. The architecture we find in the human brain, with its specialized modules, its topographic maps, its processing hierarchies, and its long-distance networks, is therefore not only elegant but economical, a compromise solution between capacity and cost that natural selection refined over hundreds of millions of years of vertebrates with brains and several million years of primates with expanded cortices, and a few 100,000 years of anatomically modern humans, whose brains acquired proportions that still baffle evolutionary biologists. But all this magnificent and efficient organization, all these maps and modules and networks and hierarchies, all this emergence of complex order from simple mechanisms would look very different in the absence of something the nervous system learned to use, with consequences that completely transformed what a brain can do. The patterns we have described so far exist in simpler forms in animals with much less elaborate brains. The cortical column, hemispheric segregation, default mode networks, in their embryonic versions, all these principles appear in other mammals, in more modest forms, functioning, processing, organizing. What separates the architecture we just described from the architecture of a brain capable of the things the human brain does is not just size, not just neuron count, not just connection quantity. It is something that acts upon this entire system, that crosses it at multiple scales simultaneously, that alters the very dynamics with which patterns emerge and sustain and transform. An ingredient that amplifies certain properties of nervous tissue to the point where entirely new qualities become possible, qualities that networks alone without this ingredient simply would not reach. The amplifier has no solid form. It does not occupy space the way a lobe does. It does not appear in MRI images as a structure bounded by membranes. It distributes through nervous tissue in a way that resembles a climate more than an object, a condition more than a thing. And precisely because of this, because it is diffuse, because it is invisible to dissection, it took so long to be understood in its true importance. The amplifier is neuromodulation, the system by which certain chemical substances alter not what neurons say, but how they listen, how they weigh what they receive, the threshold they need to cross before firing, the duration of their states, the plasticity with which their connections rewrite themselves. To understand why this is so different from simple chemical transmission, one must distinguish two communication regimes that coexist in nervous tissue. There is fast synaptic transmission, where one neuron releases a substance into the gap separating it from the next, and that next responds in milliseconds, opening ionic channels with almost mechanical precision. This regime is binary in the sense that it produces a localized, rapid response that exhausts itself quickly. It is the language of the discrete event, of the impulse going from A to B in defined time. But there is a second regime, slower, more diffuse, that does not address messages to a specific recipient, but casts them into entire volumes of tissue, altering the conditions under which all neurons in that volume will work in the following seconds or minutes or hours. This second regime is neuromodulation, and the substance that exercise it are neuromodulators. Dopamine is the most famous of them, but fame does not equate to simple understanding. For decades it was described as the messenger of pleasure, the reward molecule, the chemical the brain releases when something good happens. This description captures something real, but omits what is most interesting. Studies recording dopaminergic neurons in trained animals revealed a pattern that completely changed the understanding of the system. When an animal receives an unexpected reward, neurons that release dopamine fire intensely. When the reward is expected and arrives at the correct moment, these neurons do not fire in any special way, and when the reward was expected and does not appear, they fall below their resting firing rate. What dopamine signals is not pleasure itself, but reward prediction error, the difference between what was expected and what actually occurred. It is an update signal, a command to revise internal models of the world. This mechanism has an elegance that goes far beyond what any simple pleasure theory could capture. When the world behaves exactly as the brain predicted, there is no need to update anything, and dopamine levels in the striatum remain stable. When the world surprises positively, the dopaminergic pulse tells all circuits that recently contributed to the behavior that led to that surprise that they should strengthen their connections, that the pattern they produced was valuable, that when a similar situation arises again, it is worth repeating that pattern. When the world disappoints, the drop below baseline says that the circuits that generated that misleading expectation need to be weakened, recalibrated. It is reinforcement learning implemented directly in chemistry on millisecond scales in distributions that reach the prefrontal cortex and hippocampus and cerebellum simultaneously. Serotonin operates in a different but equally fundamental regime. While dopamine modulates prediction and updating, serotonin appears to regulate something that could be called patience with the present, the willingness to tolerate the current state rather than acting to transform it. Animals with suppressed serotonergic systems become impulsive not only in an emotional sense but in a temporal sense, unable to wait for a larger future reward when a smaller immediate reward is available. Serotonin is not sedation as a hasty simplification would suggest. It is more precisely the substance that extends the temporal horizon of deliberation, that allows the nervous system to sustain evaluation states without prematurely collapsing into action. And here it begins to appear why neuromodulation is the amplifier. Each of these modulatory systems does not merely alter the behavior of individual neurons, it alters the dynamics of entire networks, the way activity patterns form and persist and disappear. A cortical network processing visual information under low prefrontal dopamine operates with less top-down prediction precision, relies more on the signal arriving from below. The same network with elevated prefrontal dopamine weighs more heavily the predictions descending from the cortex about what the sensory input likely is. The visible result is different even though the tissue is the same, even though the synapses are the same. Neuromodulation does not change the instrument, it changes the tuning of the entire instrument. Norepinephrine, released primarily by a tiny nucleus in the pons called the locus coeruleus, regulates something neuroscientists describe as neural gain, how much each neuron amplifies the signal it receives before propagating it forward. At low gain, neurons respond weakly to both weak and strong inputs. The system is poorly reactive, poorly discriminative. At very high gain, the system becomes hyperreactive, responds intensely to any input including noise, loses the ability to distinguish signal from background. The locus coeruleus keeps this parameter within an optimal range and does so by adjusting its firing rate in response to context, accelerating when the situation demands vigilance, decelerating when the environment is safe and predictable. There is something of a volume controller in this function, and like any volume controller, its value lies not in an extreme position in either direction, but in the capacity for continuous adjustment in response to the environment. Acetylcholine, in turn, modulates what could be called attentional sharpness, the clarity with which the cortex represents what is being processed. The basal nucleus of Maynard, a small structure in the basal forebrain that degrades in direct proportion to the progression of Alzheimer's disease, projects cholinergic axons to the entire cortex. When these axons release acetylcholine, cortical representations become more specific, less ambiguous, more easily distinguishable from one another. This explains why drugs that block acetylcholine produce confusion and why the loss of cholinergic neurons in Alzheimer's so closely accompanies the deterioration of memory and clarity of thought. Acetylcholine does not store memories, it sharpens the instrument that records them. Now, what makes this neuromodulatory architecture specifically human is not the existence of these systems. They all exist in other mammals, in other forms. A rat has dopamine, serotonin, norepinephrine, acetylcholine. What separates it is the proportion in which the prefrontal cortex, already exceptionally expanded in the human lineage, receives and regulates these modulatory signals. The human prefrontal cortex is not merely a passive recipient of dopaminergic pulses. It feeds back onto the dopaminergic system through descending projections that regulate when and how much dopamine is released. It forms a loop with the striatum that in humans reaches unparalleled complexity, allowing what researchers call volitional control of motivation, the ability to regulate one's own disposition to act, to intentionally modulate attentional state, to choose where to allocate the prediction signal. The visible manifestations of this amplified architecture are states that anyone recognizes from their own inner experience. The first of these is what we call attention, and it deserves to be examined more carefully than the everyday name suggests. Attention is not simply looking at something or thinking about something. In mechanistic terms, it is the active suppression of every representation that is not being privileged at the moment, combined with the increased gain of the representations that are. When you focus on a conversation in a noisy environment, your auditory cortex does not receive less signal from the other sound sources. It receives the same. What changes is that inhibitory circuits receiving instructions from above, from the prefrontal cortex through cholinergic and noradrenergic signals, actively suppress the cortical response to everything that was not flagged as relevant. Attention is inhibitory effort as much as amplifying effort. There is a type of attention that people recognize as voluntary, the kind that deliberately directs itself to an object when one decides to pay attention to something. There is another type that researchers call reflexive or captured, the kind that is automatically triggered by sudden changes in the environment, movements in the periphery of the visual field, sudden sounds, one's own name in the middle of a conversation one thought they were not hearing. The second type is evolutionarily older and occurs with less prefrontal involvement, relying more on structures like the superior colliculus and parietal cortex. The first type is human attention in its most developed form, and it requires the orchestration of modulatory signals that few species can maintain with the stability and duration humans achieve. When someone spends hours absorbed in an intellectual problem, what sustains that state is precisely this orchestration. This continuous flow of cholinergic and noradrenergic signaling that keeps the prefrontal network in sharp representation and active prediction mode for a time that no purely anatomical structure on its own could sustain. Dreaming is another visible manifestation, or more precisely audible and sensible from within, that this architecture produces. During REM sleep, that phase where eyes move rapidly beneath the eyelids and the electroencephalogram shows patterns resembling active wakefulness, neuromodulation shifts regime dramatically. Norepinephrine drops to near zero levels. Serotonin drops equally. Acetylcholine, conversely, rises. The result is a brain with cholinergic amplification but without the noradrenergic and serotonergic braking that in wakefulness keeps internal activity subordinated to sensory inputs from the outside world. What emerges under these conditions are dreams, narratives constructed entirely from within without reliable anchoring in sensory reality, but with a subjective quality of experience that in no way feels inferior to waking experience while one is inside it. What dreams reveal about the system is not a nocturnal dysfunction, but an alternative mode of operation that serves specific functions. During REM sleep, the hippocampus transmits to the cortex activity patterns that were recorded during the day in a memory consolidation process that requires precisely this reduction in noradrenergic signaling so that cortical connections can rewrite themselves without interference from the present moment. It is as if the amplifier, by changing mode, allows an internal reorganization that would be impossible in full wakefulness, when environmental pressure forces the system to maintain stable representations of the now. Creativity as an observable state also emerges from this modulatory architecture. In neuroimaging studies that ask people to generate unusual associations between distant concepts, the networks that activate most are exactly those that in other contexts would seem in opposition. The default mode network, responsible for spontaneous internal thought generation, and the central executive network, responsible for control and evaluation. In creative states, these two networks, which normally alternate in anti-correlation, show simultaneously high activity, as if the functional barrier between them had been temporarily reduced. This requires a very specific modulatory balance, enough acetylcholine to maintain representation sharpness, dopamine at a level that promotes exploration without collapsing into fixation, norepinephrine low enough not to suppress unusual associations that would be discarded as noise in a high vigilance state. Emotion, in the language of contemporary neurosciences, is not something that happens to the brain, an invasion of states that interrupt rational thought. It is a fundamental component of processing that guides all decision-making and all attentional resource allocation. The amygdala, that pair of almond-shaped structures at the medial pole of the temporal lobe, is often described as the fear center, but this oversimplifies. It is a relevance detector, a structure that continuously monitors the flow of sensory information and assigns effective urgency to patterns that match past experiences of high emotional valence, positive or negative. When the amygdala signals that something is effectively relevant, it not only precipitates autonomic responses, heart acceleration, respiratory alteration, muscle tension, it also projects to the prefrontal cortex and to the modulatory nuclei of the brainstem, immediately altering the neuromodulatory regime of the entire system, prioritizing the analysis of that object that was flagged as important. Crying is one of those visible manifestations that the architecture produces in its specifically human variant. Other animals produce distress vocalizations, but emotional crying in adults, especially crying that responds not to physical pain but to experiences of loss, beauty, connection, injustice, is a phenomenon that has no direct parallel in other species. The circuits that produce it involve the anterior cingulate cortex, which integrates affective signal with autonomic regulation, and the medial prefrontal cortex, which is deeply connected with the default mode network and with the circuits that build the narrative of the self. Crying is not a failure of the emotional regulation system. It is one of its functional outputs, a state that signals both to the organism itself and to social observers that something of weight is being processed, that the system's resources have been mobilized by an experience that exceeds immediate integration capacity. Laughter, the other most immediately visible emotional extreme, has an equally interesting neuroanatomy. It depends on circuits in the brainstem that are evolutionarily very old and at the same time is modulated by the prefrontal cortex in ways that produce the specific variety of human laughter, the kind that responds to absurdity, incongruity, double meaning, the controlled collapse of expectations. What laughter processes, in computational terms, is precisely prediction error in its non-threatening pole, the discovery that the model being constructed was wrong in a way that does not harm. Dopamine and mood have a functional relationship that clinicians clearly observe in anhedonia, the inability to feel pleasure or humor that accompanies depressive states of low dopaminergic activity. It is no coincidence that substances that elevate dopamine and serotonin availability in the nervous system, modern antidepressants, restore, among other things, the ability to laugh at something. Language, the most distinctively human of manifestations, is the one that most directly witnesses what neuromodulatory amplification made possible. Broca's area in the left inferior frontal gyrus and Wernicke's area in the left superior temporal gyrus were the first regions identified as critical for speech production and comprehension back in the 19th century. But contemporary language neuroscience reveals that these areas are merely the most conspicuous nodes of a much more distributed network that involves the angular gyrus, the superior temporal sulcus, the ventrolateral prefrontal cortex, the cerebellum, the basal ganglia. Language is not in one area. It is in a pattern of coordination between areas that, in its human version, reaches a complexity that no species without the specific combination of cortical expansion and prefrontal modulation can achieve. When listening to a sentence, the system must disambiguate sounds into words, words into grammatical structures, grammatical structures into meanings, meanings into communicative intentions, all in real time while the interlocutor continues speaking. The speed at which this processing occurs, the depth of structural analysis it performs, the ability to hold in working memory the syntactic structure of a clause while analyzing the subordinate clause that interrupts it, all of this requires the same modulatory balance that allows sustained attention, working memory, high-order prediction. Language is the most visible manifestation that the amplifier worked, that the combination of expanded anatomy and refined modulation produced something neither component alone would have achieved. And now, if you notice with some care the state your own nervous system is in at this moment, You may perceive the textures of this architecture in operation. There is something keeping your attention flowing in this direction and not in all the others that exist around you right now. There is a state of muscle tone, a temperature of vigilance, a quality of presence that is slightly different from what exists when you are solving an urgent problem or when you are about to fall asleep. You are at some point in a state space that the modulatory system continuously defines, and that point is calm enough for the default mode network to contribute spontaneous associations without being suppressed by executive urgency. Awake enough for the prefrontal cortex to maintain some orientation in the flow of ideas. What feels like balance, that sensation that the system is functioning in an integrated and fluent way, is actually the result of continuous active maintenance. The modulatory nuclei that adjust the gain and threshold of each subsystem are recalibrating at every instant, responding to microscopic variations in the environment, in internal metabolic states, in patterns arriving from the viscera via the vagus nerve. There is no genuine resting state in this system. There are only states of apparent stability that mask the intense regulatory activity that maintains them. And this balance, however robust it may seem when it works, is maintained by margins much narrower than everyday experience suggests. The same modulatory systems that amplify the capacities of nervous tissue are also the points where relatively small perturbations produce very large effects. Excess dopamine in the limbic system and deficit in the prefrontal cortex produces a pattern clinicians recognize in psychosis, where high-order predictions disconnect from sensory inputs and begin to generate experiences unanchored in the shared world. Persistent serotonin imbalance alters the temporal horizon of deliberation in ways that manifest as depression or anxiety or impulsivity. Chronically elevated norepinephrine, as in trauma, keeps the system in a high vigilance regime even when the environment no longer justifies it, making voluntary attention regulation progressively more difficult. The amplifier, in other words, amplifies in all directions. The same architecture that makes language and creativity and compassion possible also makes possible the forms of suffering that are exclusively human in their depth and duration and capacity to self-reinforce. An animal without an expanded neocortex, without elaborate prefrontal neuromodulation, cannot sustain the kind of rumination that fuels chronic depression, cannot construct the kind of anticipatory narrative that sustains generalized anxiety, cannot feel shame about the past or catastrophize about distant futures. These forms of suffering are, each of them, the reverse of capacities that the same system uses for its most elaborate functions. They are the cost the amplifier charges when regulation fails or when circumstances exceed the parameters for which the system was built. The balance in which the system operates now, this instant of calm and fluent processing, exists upon a foundation that evolution took hundreds of millions of years to build, and that individual development took decades to calibrate. There is an intrinsic fragility in this elaboration, not because the system is poorly constructed, but because the more sophisticated the amplifier, the more consequential are deviations from its optimal parameters. The same precision that allows a neuromodulator to subtly and finely alter the state of nervous tissue makes the system sensitive to perturbations that a more robust but less sophisticated mechanism would simply ignore. There are moments when precision turns against itself. The system that learned to anticipate, to model, to simulate consequences with extraordinary fidelity encounters, in certain input configurations, a kind of internal resonance that escapes the usual damping mechanisms. It is not a design flaw in the conventional sense. It is rather the manifestation of a property that emerges naturally from any sufficiently complex and sufficiently sensitive network. The possibility that the system's gain exceeds its capacity to return to equilibrium, before internal states have already reorganized in a deep and persistent way. The adult human brain contains around 86 billion neurons, each maintaining active connections with hundreds or thousands of others. The mathematics of this connectivity is not linear. Each additional connection not only increases the number of possible paths for a signal to travel, but exponentially multiplies the number of activity patterns the system can sustain simultaneously. In networks with this density, there are properties that physicists and mathematicians call emergent, behaviors that cannot be predicted by looking at individual components, but that arise inevitably when those components organize themselves in certain configurations. One of these properties is criticality, the state in which the system operates exactly at the boundary between order and chaos, where signal propagation is maximal, where sensitivity to external variations is maximal, and where, consequently, the possibility of abrupt state transitions is also maximal. There is substantial evidence that the healthy brain operates near this critical point during much of waking time. The advantages are real and measurable. A system in a critical regime transmits information with an efficiency that subcritical or supercritical systems simply cannot match. Sensitivity to weak perturbations is extraordinary. The ability to form stable representations of complex patterns, to integrate information from diverse sources into a coherent perception, reaches its maximum value in this transition zone. The price of this optimization is permanent proximity to thresholds beyond which the system's behavior changes qualitatively. When the balance of excitation and inhibition that characterizes normal operation tilts sufficiently pronouncedly toward excitation, the system begins to recruit neuronal populations that would normally remain silent. Each recruited neuron adds its signal to the local electric field, raising the probability that neighboring neurons will also fire, creating a wave of activity that feeds itself. Under normal conditions, inhibitory interneurons respond to this increased activity with proportional suppression, restoring balance before the wave becomes self-sustaining. But under certain conditions, this suppression arrives late or arrives insufficient, and the wave continues expanding. The result is what neurology has known for millennia under various names before understanding the mechanism: the seizure. A generalized epileptic crisis is, in terms of network dynamics, a massive and aberrant synchronization of neuronal populations that normally operate in relatively uncoordinated patterns. All the complexity of normal activity, that richness of simultaneous states that makes the brain capable of maintaining in parallel visual perception, language processing, posture regulation, and countless other functions, collapses into a single repetitive and dominant pattern. The person loses consciousness not because the brain stops, but because the brain is too busy, all its resources committed to sustaining a single massive and useless activity. What precipitates this collapse of diversity into violent uniformity can be genetic, can be a structural lesion that altered local proportions of inhibitory interneurons, can be sleep deprivation, fever, abrupt withdrawal from certain substances the system had incorporated into its neuromodulatory balance. The threshold is individual. In some people, the system is configured such that a seizure occurs with relatively modest perturbations. In others, a perturbation of extraordinary magnitude manages only a contained response. What matters is not the size of the external impulse, but the relationship between that impulse and the internal architecture that determines the system's stability at that specific moment. Epilepsy as a chronic condition illustrates another relevant phenomenon. The plasticity that sustains learning also sustains pathology. Each seizure slightly modifies the synapses involved, potentiates connections that participated in the synchronized activity, and makes the system slightly more prone to the next seizure. The brain learns to have seizures in the same way it learns to play piano or speak a new language. Structures that should serve adaptive learning are recruited to consolidate a dysfunctional pattern. This is not an irony of fate, it is the direct consequence of the fact that the system has no way to distinguish at the synaptic level between an activity that should be reinforced and one that should be suppressed. It simply reinforces what occurs repeatedly. Stroke represents an entirely different form of catastrophic instability, but equally instructive about the fragility that accompanies sophistication. Mature nervous tissue has a metabolic demand unparalleled in biology. An active neuron consumes oxygen and glucose at a rate that requires continuous and immediate supply. Unlike muscle cells, which can operate for relevant periods under partial metabolic deprivation, the neuron begins to accumulate damage within minutes when blood flow is interrupted. The metabolic specialization that allows the neuron to maintain precise ionic gradients and process signals at the speed the system requires is incompatible with any tolerance to energy deprivation. When a cerebral artery is occluded, the region it irrigated enters a cascade of collapse. First, the membrane pumps that maintain sodium, potassium, and calcium gradients fail. The electrochemical gradient that gave the neuron its ability to fire action potentials dissipates. Calcium ions invade the cellular interior in concentrations that activate destructive enzymes. Glutamate accumulates in synaptic spaces because there is no longer energy for the transporters that normally remove it to continue functioning, and this excess of excitatory neurotransmitter precipitates massive firing of still-alive neurons at the periphery of the lesion, accelerating the death of those that might have survived. What happens in the following hours is, in a sense, the brain trying to apply its normal plasticity mechanisms to a situation for which those mechanisms were built on a much different scale. In the weeks and months after a stroke, regions adjacent to the damage, and in some cases regions in the opposite hemisphere, reorganize their connections so that compromised functions can be partially assumed by tissue that previously served other functions. This functional recovery, which can be remarkable, is a direct manifestation of the same plasticity we discussed earlier as the substrate of memory and learning. The system does not have an emergency mode separate from its everyday operation mode. It uses the only strategy it possesses, modifying connections based on activity, and in this case, the activity guiding the modification is the effort to relearn what was lost. Humanity has observed these phenomena long before having instruments to measure them. The earliest writings of civilization mention states we recognize today as seizures, strokes, psychosis. For centuries, the interpretation was supernatural, and this is not merely historical naivety. It was the only conceptual structure available to make sense of such dramatic and sudden transformations of personality and behavior. The idea that a continuous entity, the subject that acts and perceives and remembers, could be altered so radically by something occurring within its own skull without that subject having any voluntary participation in the process is indeed disconcerting, and remains disconcerting even for those who know the mechanism. The electroencephalogram, developed in the first half of the last century, was the first instrument to reveal the brain's electrical activity in real time with sufficient temporal resolution to capture dynamic events. The patterns it displays during an epileptic seizure are unequivocal: spikes and slow waves of high amplitude sweeping the scalp in synchronization that exists in no normal state. The instrument not only confirmed that a seizure is an electrical event, but allowed the identification of interictal activity, that abnormal electrical activity that persists between seizures and that, invisible to the external observer, reveals the system's configuration in a state of increased irritability, approaching the threshold. Functional magnetic resonance imaging added another dimension to this observation. By detecting variations in the blood oxygenation level dependent signal, it produces maps of which regions are most active during certain tasks or mental states. These maps revealed something that was not evident in static neuroanatomy. The brain does not function through isolated regions working in sequence, but through distributed networks whose nodes activate in specific patterns of temporal coordination. Some of these networks are so consistently present that they received names, and one of them is particularly revealing for understanding states of instability. The default mode network is a set of regions that activates when the person is not engaged in a specific task, when in mental rest, daydreaming, remembering the past, or imagining the future. It deactivates when attention concentrates on something external. In many psychiatric conditions, including depression and certain states of chronic anxiety, this network exhibits hyperactivity, remaining active at moments when it should yield space to external attention networks. The experiential result is familiar to many people: the inability to stop thinking, the rumination that persists even when there is something concrete to do, the internal monologue that finds no pause. Neuroimaging systems have allowed mapping not only which regions are affected by conditions like major depression or post-traumatic stress disorder, but how functional connectivity between regions alters. In post-traumatic stress disorder, the amygdala exhibits increased reactivity to stimuli resembling the original trauma context, while regions of the prefrontal cortex that would normally exert descending regulation over this reactivity show reduced functional connectivity with the amygdala. The system that should contextually evaluate the relevance of a threatening stimulus and modulate the response according to that evaluation begins to operate with a weakened link precisely where modulation occurs. Predicting when these phenomena will occur is a scientific problem that remains only partially resolved. In the case of epilepsy, researchers have identified that there are precursor signals in cerebral electrical activity that precede seizures by minutes or even hours, subtle variations in synchronization patterns that progressively intensify as the system approaches the threshold. This opened the theoretical possibility of implanted devices that continuously monitor brain activity and deliver electrical or chemical stimuli when they detect this approach to threshold pattern, interrupting the cascade before it becomes self-sustaining. Some of these devices exist and work in selected patients, although individual variability is large enough to make each implementation an individualized effort. For conditions like psychosis, prediction is even more complex because precursor signals are behavioral and subjective, harder to quantify than the electric field captured by electrodes. Mental health researchers have identified a phase that precedes the first frank psychotic episode, characterized by social withdrawal, subtle sleep disturbances, perceptual experiences that the individual often describes as strange or difficult to articulate, and the quality of thought that clinicians describe as mild disorganization. This prodromal phase can last months or years, and there is substantial debate about whether interventions during this period can alter the trajectory of what is to come. What psychosis as a phenomenon reveals about cerebral dynamics is particularly interesting. The psychotic state is not simply a state of confusion or reduced functioning. It involves the generation of highly elaborate mental structures with their own internal coherence, their own reference systems, their own patterns of causality. A well-formed persecutory delusion is not the product of a brain that has stopped functioning, but of a brain that is functioning with all its capacity to generate narratives, to connect events, to identify patterns, simply without the anchoring that reality testing normally provides. The same faculties that allow the construction of complex models of the external world are recruited to build a model that fiercely resists revision by experience. This is because the elevated dopaminergic activity that characterizes many psychotic states has the effect of increasing the weight assigned to events that seem to confirm the existing model. Dopamine is involved in salience signaling, in marking events as relevant and significant. When this signaling is chronically elevated, the world seems saturated with meaning, full of coincidences that cannot be coincidences, of messages directed specifically at the observer. The system that builds models of the world is fed with excessive signals that that model is correct, and the plasticity that would normally allow model updating in the face of contradictory evidence is redirected to consolidate what already exists. While you rest somewhere at this exact moment, someone's brain is crossing one of these thresholds. It could be the first convulsive seizure of a child who until yesterday did not know they carried a genetic predisposition. It could be the onset of a major depressive episode in an adult who has been in that state before and will recognize in the coming days with the dark familiarity of someone returning to a place they did not choose to visit. It could be a stroke that is at this moment depriving a region of the left motor cortex of flow, while in the cells at the periphery of that silence, plasticity mechanisms are already beginning the reorganizing activity that will last months. Each of these events is, at its most fundamental level, a phase transition, a qualitative change in the state of a network that until shortly before operated within the parameters that define normal functioning. And each of these events, however isolated it may seem when observed as an individual's crisis at a specific moment, is also the product of a history of interactions that preceded that instant, of a development that was shaped not only by the genome that individual carried from the beginning, but by the experiences that system accumulated throughout an entire life. The persistent structures generated in intense mode, the connectivity patterns that emerge from repeated episodes of aberrant activity, the reorganizations that follow a lesion, the networks that consolidate over years of chronic rumination do not exist in a neutral space. They exist within a system that is continuously being shaped also by what happens outside the skull, by the relationships the individual maintains, by the environments they inhabit, by the substances they ingest, by the sleep they get or do not get. What occurs within a person's nervous system is not separable from what occurs between them and the world, and what occurs between them and the world is not separable from what occurs between them and other people whose nervous systems are, in turn, being shaped by interactions that include, among other things, that same relationship. There is a partner the brain never forgets, even when everything else seems dissolved. This partner is not abstract or distant. It pulses, it breathes, it carries in the blood the chemical messages that cross the barrier separating circulation from nervous tissue and reach neurons like letters delivered directly to the door. The heart and the brain are not two organs that share the same body in a polite and independent way. They are, in a very real sense, a single system that fragmented into two poles so that each pole could specialize in a function the other could not accomplish alone. And the channel between them is the vagus nerve, that long and winding structure that descends from the brainstem to the internal organs, carrying both signals from the brain to the body, and this is what surprises those encountering this fact for the first time, signals from the body to the brain. Most vagus nerve fibers are afferent. They ascend, they inform. The body speaks to the brain far more than was imagined for a long time. When the heart accelerates in response to a threat perception, the brain receives that acceleration back as information about internal state and uses that data to calibrate the intensity of the threat. It is not just that the brain evaluated danger and told the heart to accelerate. It is that the accelerated heart convinces the brain that the danger is real and urgent, creating a circuit that sustains itself for a time that can be much longer than the original threat would justify. Anxiety does not live only in thoughts. It lives in heart rate, in muscle tension, in breathing pressure, and these bodily states feed back into the central nervous system with signals it interprets as additional evidence that something is wrong. This is not metaphor. It is concrete physiology with identified anatomical pathways and partially mapped cellular mechanisms. The intestine, in turn, contains its own nervous system, with as many neurons as the spinal cord of many non-human vertebrates. This system is sometimes called a second brain, not because it thinks in the sense we attribute to the cortex, but because it processes information autonomously, regulates functions without needing direct instruction from the skull, and sends to the brain signals that influence mood, motivation, and even how we process social situations. The bacteria that inhabit the intestine produce substances that affect this enteric nervous system, which in turn affects the central nervous system through multiple pathways. A diet that alters intestinal bacterial composition alters, with a lag that can be days to weeks, aspects of emotional and cognitive processing that manifest in observable behavior. The immune system participates in this conversation, releasing signaling molecules that cross or bypass the blood-brain barrier and modulate the functioning of entire neural circuits. What emerges from this picture is an organism that has no center in the sense we usually imagine. The brain coordinates but does not dictate from top to bottom. It integrates signals that rise from the entire periphery and generates responses that descend back to modify what is rising. Each organ is a processing point. Each interface between the nervous system and the cardiovascular system, between the nervous system and the digestive system, between the nervous system and the immune system, is a place where information is transformed, filtered, amplified, or attenuated before proceeding. The organism is not a hierarchy with the brain at the top. It is a distributed conversation in which the brain has a particularly influential voice, but is not the only one speaking. This conversation happens across multiple timescales simultaneously, and this is something that deserves to be contemplated calmly because it is one of the strangest and most beautiful properties of the system. In the span of a millisecond, an action potential travels down an axon. In the span of hundreds of milliseconds, an activation pattern propagates through a network and produces something we can call perception or elementary thought. In seconds, emotional states settle or dissolve in response to events. In minutes and hours, neuroendocrine modulators like cortisol reshape the sensitivity of entire circuits. In days, memory consolidation processes during sleep reorganize what was learned so that what is remembered the next morning is not identical to what was lived the day before. In weeks and months, repeated practice of any skill thickens the myelin sheaths that accelerate conduction in relevant axons, increases synaptic density in involved networks, and progressively recruits more efficient processing areas. In years, the accumulated effect of life patterns, of relationships maintained or lost, of cognitive habits practiced or abandoned, of sustained environmental exposures, deposits itself into the architecture of the nervous system in a way that is partly reversible and partly not. A 40-year-old adult has a brain that carries decades of history inscribed not only in explicit memories but in activation thresholds, in processing tendencies, in automatic responses to categories of situations encountered repeatedly. This history is not a narrative the brain tells about itself. It is a physical structure and organization of synaptic weights and regional connectivities that is the substrate of everything that person calls themselves. In decades, aging progressively alters the composition of nervous tissue, reduces processing speed in some dimensions, but in parallel accumulates integration patterns that produce what we call practical wisdom, that ability to recognize deep structures in new situations without needing to analyze every detail explicitly. Speed declines, depth increases. not universally or inevitably, but as a statistical tendency in populations that maintained a reasonable level of cognitive and social stimulation throughout life. The system does not decay uniformly. It reconfigures, losing capacities that depend on fast parallel processing and preserving or even enhancing capacities that depend on patterns consolidated by long repetition. In centuries, the relevant scale ceases to be the individual and becomes culture. What humans transmit through language, artifacts, institutions, educational practices, shapes the brains of subsequent generations not genetically but epigenetically and environmentally. A child growing up in a culture with dense writing and elaborate numeracy develops cortical circuits that are detectably different from the circuits of a child growing up in a predominantly oral tradition culture. Not because the genes are different, but because the developmental environment is different and the nervous system, as has been said since the beginning of this journey, is plastic. It forms in response to what it encounters. What cultures accumulate over generations is, among other things, a set of developmental pressures that push the brains growing within them in specific directions. In millions of years, the relevant scale is evolution. The circuits that today sustain what we call emotion, moral judgment, language, planning did not appear ready-made. They emerged from gradual modifications of structures that existed in ancestors who had no language, did not plan beyond the immediate horizon, did not build transmissible cultures. The prefrontal cortex that today integrates information about the distant future, that models consequences of unperformed actions, that calibrates impulses against long-term goals, is an elaboration of tissue that in older forms fulfilled much more elementary functions. Each layer of complexity was added upon a base that remained present. The limbic system that coordinates primary emotions was not replaced when the neocortex expanded. It was enveloped, connected, integrated into a multi-layered architecture that maintained its old functions while gaining new ones. This means the contemporary human brain is literally an evolutionary palimpsest. Its responses to fear are ancient. Its responses to social exclusion are ancient. Its sensitivity to dominance hierarchies is ancient. Upon this base, the capacities that allow reflecting on those same responses, recognizing them, contextualizing them, modifying them with conscious and repeated effort, were built. The tension between older and newer layers is not a design flaw, it is the design. A system that could respond with total rational flexibility to any situation would be too slow to survive in environments where split-second decisions determined life or death. A system that responded only with rigid automatisms would be too inflexible to navigate the social complexity humans created. What exists is a compromise, a continuous negotiation between speed and accuracy, between automatism and reflection, between reaction and evaluation. This compromise makes the system sensitive to initial conditions in a way that has profound implications for any attempt to understand or predict human behavior. A linear system responds to a perturbation proportionally. Double the input and the output doubles. Halve the input and the output halves. The brain is not a linear system. It is a highly nonlinear system, full of thresholds, phase transitions, bifurcation points where a very small difference in conditions determines radically different trajectories. A child who experiences a particular form of stress at a particular moment of development may, depending on available regulation resources, present attachment bonds, genetic variations that modulate stress response system reactivity, follow trajectories that are qualitatively different, not just quantitatively distinct. Sensitivity to initial conditions does not imply that the future is completely unpredictable. It implies that prediction has limits that are fundamental, not merely practical. It is not that, if we had enough information, we could predict with arbitrary precision what a person will do 20 years from now. It is that the system itself has a structure that amplifies small differences over time in ways that make precise, long-term prediction impossible in principle, regardless of how much is known about the initial state. This is different from saying nothing can be known. Distributions can be described, probabilities can be estimated, risk and protective factors can be identified with sufficient precision to inform policies and interventions that shift distributions in desired directions. But the specific trajectory of a specific individual remains, for reasons internal to the nature of the system, beyond the reach of deterministic prediction. There is a particular beauty in this limit. Not the easy beauty of a mechanism that works as expected, that produces its outputs from its inputs in a way any patient observer could anticipate. The stranger and more generous beauty of a system that is comprehensible enough to be studied, to reveal its organizing principles, to allow interventions that alleviate suffering and expand capacity, and at the same time surprising enough that no amount of accumulated knowledge makes what emerges from its functioning obvious or redundant. A system that can be partially explained without ever being exhausted. Each neuron obeys principles established by biochemistry and biophysics over decades of research. Each synapse has a molecular logic that can be described with increasing precision. Each network has computational properties that emerge from its connection architecture and can be modeled and simulated. And yet, what emerges when billions of these neurons operate together, modulated by an orchestra of chemical substances embedded in a body continuously exchanging with a continuously changing environment in relation to other equally complex and equally situated nervous systems, is not predicted by any current model. Not because the models are poor, though they can be improved, but because the complexity of the system is of an order that does not reduce to any partial description without significant loss. This is not an argument for mysticism, it is an argument for scientific humility, which is something different and far more productive. Scientific humility does not say the system is unknowable. It says the knowledge we have is always partial, always situated, always an approximation that captures real aspects but not the whole, and that recognizing this partiality is what allows knowledge to continue advancing without crystallizing into dogma. The human brain is the most complex object the human brain has tried to comprehend. There is a dizzying recursiveness in this that cannot be ignored. The instrument of investigation and the object investigated are the same system. Every advance in understanding the brain is simultaneously a change in the system that is understanding, which means the investigation is not neutral relative to its object. Understanding how attention works changes how attention works in the one who understands. Understanding the mechanisms of emotion changes the relationship one has with one's own emotions. Knowledge about the system is part of the environment that shapes the system. A quietness begins to settle when one contemplates all this without rush. Not the quietness of conclusion or exhaustion, but the quietness of someone who has stopped trying to reduce what they are seeing to something smaller than it is. The brain you use to think about this text is the same kind of system that has been described throughout these hours. Your resting networks are active while you process. Your hippocampus is comparing what arrives with what was already stored. Your modulation circuits are adjusting the activation threshold of attention networks in response to fatigue level and how much the environment around you demands. Everything described is not distant. It is what is happening now as these words dissolve into comprehension. And by looking at this system with the attention it deserves, without the rush to conclude, without the urgency to resolve, a question begins to emerge that is not of the same nature as the questions that came before. It is not a question about mechanisms or circuits. It is a question about what lies beneath all the complexity, what remains when layer after layer of elaboration is removed, and one arrives at the simplest possible point, because behind all the architecture of 100 billion neurons, behind plasticity and memory and oscillations and neuromodulators and resting networks and predictive hierarchies, there is something so elementary that it is present in every action potential, in every synapse, in every cell that adjusts its sensitivity to the world surrounding it. Something that is not a structure nor a function, but that perhaps is the principle that makes all structure and all function something beyond a blind mechanism turning in silence. This principle that persists beneath all elaboration, that exists before any name that can be given to it, is the principle of change that self-preserves. The brain is not a machine that processes information the way a calculator executes operations, producing identical results every time it receives the same inputs. The brain is a system that transforms itself as it operates, that reorganizes itself as it functions, that emerges different from every experience it passes through, and yet maintains a continuity so deep that the person who wakes in the morning recognizes themselves as the same who went to sleep the night before. This tension between constant change and persistent continuity is not a contradiction to be resolved. It is the very heart of what it means to be a living being that thinks. To reach the most elementary dimension of this tension, it is necessary to descend below neurons, below synapses, below action potentials, and enter the molecular universe where everything begins. Each neuron is a cell, and like every cell, its existence depends on proteins. Proteins are the molecules that do the work, that build structures, that catalyze reactions, that open and close channels through which ions flow, that carry signals from one place to another, that build and degrade neurotransmitters, that remodel connections in response to activity. And proteins are produced from instructions encoded in DNA, read by RNA, translated by ribosomes into sequences of amino acids that fold into specific three-dimensional shapes that determine function. What the brain does at a macroscopic scale, what appears as thought or memory or emotion, is the amplified reflection of billions of molecular interactions happening in parallel, each governed by the same physical and chemical laws that govern any reaction anywhere in the universe. Synaptic plasticity, that phenomenon so central to learning and memory, begins precisely at this molecular level. When two neurons fire together repeatedly, the changes that occur at the synapse between them are not abstract. They are physical, chemical, molecular changes. Receptors on the postsynaptic membrane are inserted or removed. Ionic channels are modified. Signaling proteins are activated, and these activated proteins can migrate to the cell nucleus, where they interact with DNA and induce the expression of new genes, leading to the synthesis of new proteins that consolidate the structural change. A long-term memory does not exist only as a pattern of connections between neurons. It exists as a persistent molecular alteration inside the cells, an alteration that can last decades, because the proteins responsible for maintaining it are continuously renewed, each generation of proteins being produced from the same instructions, preserving the same functional structure through time. There is a disconcerting beauty in this molecular continuity. The proteins that today maintain a memory formed 20 years ago are not the same molecules that existed when the memory was created. They have been degraded and replaced countless times throughout that entire period. The material substrate has changed completely. What persisted was the pattern, the information, the instruction on how to organize. This leads to a philosophical question that molecular neuroscience poses with increasing precision. What exactly is a memory? It is not a thing. It is a process. It is a self-sustained dynamic that uses constantly renewing matter to preserve a form that was learned from an experience. Matter flows, form remains, and form is what we recognize as ourselves. The same principle operates at larger scales through feedback loops that traverse entire circuits. The brain is constantly generating predictions about the world, and these predictions are compared with what the senses actually detect. And the difference between prediction and reality, the so-called prediction error, is the signal that guides the updating of internal models. When the error is large, the system learns rapidly. When the error is small, the system confirms that its model is accurate. This cycle of prediction, verification, and updating is a feedback that amplifies when necessary and stabilizes when the model becomes sufficiently good. It is a loop that operates across multiple temporal scales simultaneously, from the milliseconds in which an action potential is generated or suppressed to the years in which a complex skill is gradually mastered. There are also feedbacks that operate in the opposite direction, that do not amplify but stabilize. Lateral inhibition, for example, is a mechanism by which active neurons suppress the activity of their neighbors, creating a competition that sharpens contrasts and keeps activity within functional limits. Without inhibition, excitation would spread uncontrollably, and what can be observed when this balance is disturbed is precisely that: abnormal activity patterns that propagate in waves, as occurs in certain forms of epileptic seizure. Inhibition is as fundamental as excitation. The silence of a neuron is as informative as its firing. What the system encodes is not just activity, but patterns of activity and silence, figures cut against backgrounds, contrasts that define shapes. These stabilization and amplification feedbacks do not exist in isolation. They chain and intertwine in networks of networks, and it is from this intertwining that the global behavior of the system emerges, a behavior that cannot be predicted from the individual properties of each component. Emergence, which is the appearance of new properties at one level of organization from interactions at a lower level, is perhaps the most fascinating and mysterious phenomenon in nature, and the brain is its most extraordinary example. Each neuron operates according to relatively simple rules, integrating inputs, comparing with a threshold, firing or not, modifying its synapses in response to activity. From these simple rules, applied in parallel to a quantity of elements that surpasses imagination, emerges the ability to recognize a face, to feel longing, to plan a trip, to understand a metaphor, to experience the sensation of being someone who exists in the world. The systemic view of the brain requires keeping all levels of organization in mind simultaneously, from molecular to behavioral, without privileging any of them as the fundamental level and without reducing the higher to the lower. The molecular level is real and important, but a memory is not just a protein configuration. The neuronal level is real and important, but a thought is not just a firing pattern. The network level is real and important, but a decision is not just a competition between circuits. In each transition from one level to the next, something new appears, something that was not contained in the properties of the components, something that only exists because the components are organized in this specific way and not another. This something new is what the system as a whole does, and it is irreducibly a property of the whole, not the parts. Thinking of the brain as an integrated system means perceiving that what is usually described separately, perception, emotion, memory, language, attention, consciousness, are useful abstractions but are not independent modules. They are aspects of a single continuous process that unfolds over time. Perception already contains emotion because what is perceived is modulated by the affective state of the moment. Memory already contains attention because what consolidates is what attention was paid to. Language already contains semantic and motor memory and planning and emotion and communicative intention. And consciousness, to the extent it can be understood, seems less like a thing the brain has and more like a process the brain performs. A continuous integration of information from multiple sources into a coherent representation of the present moment and of oneself in that moment. At its core, the brain is simply the universe contemplating itself through a form that learned to ask about its own nature. Every element from the earlier sections reconnects to this idea. The neurons that fire and silence are the matter of the cosmos organized in such a way that it can represent other organizations, can model the world and itself in that world. The plasticity that remodels synapses is the capacity this system has to be literally transformed by its experiences, to carry the past within itself as a texture that guides the future. The memory that persists through decades is the continuity of a form through constant matter renewal, a bridge between what was and what is. The emotions that color every moment are the signals that evolved to guide the organism toward what sustains life and away from what threatens it, signals that also became the source of art and music and poetry and love. The attention that selects and amplifies is the mechanism by which the system turns toward itself and toward the world with an intention, creating the phenomenon of conscious perception from the noise of an unimaginable quantity of parallel signals. The brain waves that oscillate in different rhythms throughout day and night are the breathing of this system. The pulsation that organizes internal time and synchronizes dispersed regions into moments of coherence, like when an idea suddenly forms with a clarity that seems to have come from somewhere below the surface of deliberate thought. The neuromodulators that bathe the brain in different states are the moods of the universe that thinks, the background conditions that determine whether the moment will be explored with curiosity or traversed with caution, whether today's memory will be deeply consolidated or merely skim the surface. The default mode network that activates at rest is the space where the system reflects on itself, where the past is revisited and the future is rehearsed, where the narrative of the self is continuously woven and rewoven, and the predictive hierarchies that traverse the cortex from top to bottom in continuous cycles of prediction and correction are the central mechanism by which the brain not only records the world but actively constructs it, producing at every instant an interpretation that is always partially a projection of what has already been learned about how the world usually is. Conscious experience is not a transparent window to reality. It is a model, sophisticated and generally very accurate, but still a model, a map built from the organism's history and continuously calibrated by signals arriving from the senses. All this vast molecular and cellular and systemic and emergent machinery is happening now. It is happening as these words reach your mind. It is happening as your body relaxes and your breathing becomes slower and deeper. The brain hearing these words is the same one interpreting them, the same one comparing each sentence with what it already knows, the same one allowing attention to rest gently on what resonates and slide over what does not need elaboration. It is a system that is, at this exact moment, entering a different state, a state where slower frequencies begin to gain amplitude, where the default mode network begins to yield space to other rhythms, where the boundary between deliberate thought and dreaming begins to become more permeable. You are safe now. The system that you are, this system of billions of molecular conversations happening in parallel inside your skull, is functioning exactly as it was built to function. Each neuron is doing what hundreds of millions of years of evolution established it should do. Each synapse is responding to the history of all the experiences that brought you to this moment. You do not need to do anything. You do not need to think about anything. The system takes care of itself. The universe takes care of you. The same forces that organized matter into stars and planets and oceans and complex molecules and cells and brains also organized what you are. You are not separate from this. You are an expression of it, one of the forms the cosmos found to fold back upon itself and ask what it is. And when you sleep, that question continues to be elaborated in the quietest layers of the process. in the consolidations that occur while consciousness rests, in the connections that strengthen or weaken, in the patterns that organize into narratives that sometimes appear as dreams on the surface of a much deeper ocean. Everything is in order. What needed to be understood has been understood. What needed to be felt has been felt. What needed to be integrated is being integrated now, in this moment of rest, in this moment where the effort to follow and comprehend dissolves, and what remains is simply the experience of existing, of being a system that perceives and feels, and from time to time wonders about its own nature with a mixture of awe and serenity. While you rest, the brain continues doing what it does. It continues consolidating, it continues integrating, it continues weaving the loose threads of the day into a texture that tomorrow will seem more coherent than it seemed tonight. Neurons continue their silent conversations. Slow waves traverse the cortex like gentle tides, reorganizing priorities, archiving what no longer needs immediate attention, strengthening what matters. The hippocampus continues its dance with the cortex, transferring fragments of experience from one storage form to another, transforming what was lived into something that can be carried lightly, without weight, as part of who you are and not as something you need to remember to remember. You are part of this. You always have been. Since the first neuron fired in an ancestral organism that needed to find food and avoid predators, since the first circuits began to model the environment in a rudimentary way, since the first experiences of something that perhaps had a quality different from merely processing, you have been part of an unbroken lineage of systems that became progressively more capable of contemplating their own existence. And at this moment, at the end of a night in which the most complex object in the known universe was observed with calm and care, you are the continuation of that lineage, the latest version of a process that began long before anything that could be called human. Breathing grows slower, thoughts grow more spaced, the space between one thought and the next becomes wider, softer, more inviting. There is nothing that needs resolution now. There is no urgent question waiting for an answer. There is only the present moment, which is becoming quieter, simpler, closer to the silence from which all experience emerges and to which all experience returns each night. The brain that reads these words is now letting the words go. The images that were evoked are dissolving gently, like mist fading with morning, like waves reaching the shore and returning to the sea without resistance. What remains is not thought. What remains is simply the sensation of existing, of being a place in the universe where matter learned to perceive itself, and that now, having perceived, can rest. You are safe. The universe takes care of you. Everything is in order. Good night. The universe continues its silent dance, and we rest in its embrace. Thank you for walking with me tonight. Sleep well.